Hello, everyone. Thank you for the invitation, the organizing committee. It's an honor to be here, and especially it's an honor to represent my institution, Fundecitrus, where I have been working for the 23 years already. And I will present what we planned for today. I will make a short description of the Brazilian citriculture very shortly, and then I will explain how Fundecitrus operates. I will explain about our big threat, that is a disease that was mentioned already yesterday, that's called greening or Huanglongbing. And at the end, I will go a little bit deeper into the biotechnology program that we have to overcome this important disease. Okay, the Brazilian citriculture, I will present some overall numbers. You have seen very big numbers in yesterday and today. And citriculture, it's a little bit smaller than the other cultures, but with the land that we use, the oranges that we have in fields and the oranges that we produce, we are the largest orange producer worldwide, and we produce the higher amount of orange juice comparing with the other countries. For each 10 orange juice consumed worldwide, 8 are produced in Brazil. So citriculture industry is very important in Brazil. It's cultivated along the country, but most of the oranges are grown around São Paulo state and Minas Gerais state. So we are in the belt of citrus orange production close by here. So, for instance, Fundecitrus is located 1 hour from here. It's our center, is our headquarters, located in Araraquara. Fundecitrus was founded almost 50 years ago. It's a non-profit association. We are kept with a budget comprised by a box tag, a voluntary box tag. This is important. From each box of orange that goes to the industry, some cents are collected from growers or from industry, and that's make our budget to keep our activities. And we work for all the sector, all citriculture in São Paulo and Minas Gerais, and where the citriculture is expanding in the last few years, we are working closely, very closely with the growers. And that's our mission, that is our purpose, to help growers to keep the production in very good condition and in safe condition and healthy conditions. Fundecitrus was created due to citrus canker, a disease that was spreading in the 60s, 70s, and was making danger for the production. So along these 47 years of experience, several outbreaks came, and the last very important outbreak came in 2004, that is named greening. So Fundecitrus partnered with different research institutions across the world, across São Paulo and Brazil state. We have public and private financing for our activities, and we work in several areas. I will not go in detail. I invite those who doesn't know Fundecitrus to go to our website and see our main activities. I will go into details about how this disease that came from Asia, citrus came from Asia. All citrus that we consume, orange, limes, lemons, they came from Asia. So they are the cultivated citrus. But also from Asia came some problems, like the psyllid vector that came more than 60 years ago to Brazil, and 21 years ago, the bacterium that caused HLB. HLB stands for Huanglongbing, the name of the disease in Chinese, the formal name, and we call it also greening, is the worldwide threat for citriculture. It is spread across Americas. It's spread in Asia, South Africa. It's not present yet in the Mediterranean area, although they have the vectors, and it's not present in the Oceanian region. So this is the big threat for the industry currently. We have about 44% of the trees affected by HLB, symptomatic trees. And just for a comparison, in Florida, that was a big partner in orange juice production. The production dropped more than 90% in the last 20 years due to HLB mainly. So this is the big problem we have. And to help the growers to cope that, we launched 17 years ago one biotech project, along with different projects that we have for growers. There was a report in Vietnam that guava trees, when intercropped with citrus trees, they reduce the incidence of greening in field conditions. And that stands for at least 2 years. And when the citrus trees grow more than guava trees, those effects were not present anymore. So Fundecitrus did a mission in 2008 to understand that in field conditions. The purpose was not to bring the idea of intercrop to Brazil, because we plant citrus in very high scale, but to understand the situation, what was going on there, and take advantage of that information. So one of the understandings that we did with our partners was that there was a volatile from guava leaves that ourselves we cannot smell, but the psyllid dislike. The psyllid dislike this smell when we do experiments with olfactometry. So the psyllid goes to the opposite direction of this smell. So this was an information that guava leaves, they have some repellent volatiles. When we compare the volatiles profile from orange and from guava, they are distinct. They are similar in the main content. They are terpenes. You all know that orange produces limonene, for instance. And guava, they emit another terpene called caryophyllene that we will call now on repellent. So this compound was the main candidate to be the repellent, and when we put this compound alone in olfactometry assay, it repels the psyllid. When we engineer a model plant called Arabidopsis thaliana to produce much more of this compound, the plant itself repels the psyllid, which is, this plant is not a host for this insect, but the plant became repellent for the psyllid. So with that information, we have two basements for our biotechnology project. At the same time, we figure out that sweet orange trees, they have this compound, this caryophyllene compound, in tiny amounts in the leaves. So we designed a strategy, a biotech strategy, to add a gene on sweet orange and increase this amount of this compound. I will not have time to explain in details, but I will summarize in the next steps 8 years of research in laboratory and greenhouse conditions, where we generated several new lines of sweet orange, Pera sweet orange, Valencia sweet orange, and we selected for the presence and activity of the genes, the content of the molecule of interest, the repellent, the emission of this repellent, and the behavior of the psyllid. Because in the field conditions, you have psyllids flying away, and when they fly and go to a healthy tree, they feed on the healthy tree, and they infect the healthy tree with the bacterium. And you cannot cure this disease. The only solution for this is to remove that infected tree, to reduce the source of inoculum for new trees to be infected. So this is a disease. It's a disease that spreads along the country and affects all growers. So along these years, we selected several lines that are very highly producer of this compound, and we tested in laboratory conditions, greenhouse conditions, but at the end, we have to plant a field, and we planted that 6 years ago to see the behavior in natural, in agronomical conditions, and to do that, we selected an area, 30-hectare area, and we plotted these plants in different formats, different constructs, and we assessed along these 6 years the incidence of infected trees, the incidence of the tiny insect that spread the bacterium in yellow sticky traps attached in the leaves, and several agronomical parameters for the plants. This is an area plot of the experiment. For instance, each parcel, it's about half of the size of this room. We call a large parcel because we have to see the epidemiology of the disease, and we have also small parcels to compare the importance of the size. The plants grow, they produce flower, fruits. The fruits have juice, similar as the control ones. And at the end, we observed that the original Valencia trees, they have double amount of infected trees than the repellent trees that you produced. Meaning that in field conditions, we developed a new biotech trait that was not present in other crops. We could not take advantage of the biotechnological developments from other crops. We have to raise a project from the scratch, from the original idea, where we can have at least a reduced level of the disease along the cycles of the plants. We also observed much less psyllids in the yellow sticky traps, meaning that the plants are repelling the psyllid. Okay? This along 5 years in a row in field conditions. But this strategy is not a resistant tree. It does not avoid completely the disease. So we need to work closely with the management strategies that growers are using currently. So when we provide this information, we are in the middle between having this information for growers and for the consumers. We are in the middle of our research project. But when we apply this in field conditions in the future, and we call the growth of the future, we will combine the biotech strategies with the management strategies that the growers were helped to develop using biological control, insecticides, systemic insecticides, cowling that avoids also the feeding of the psyllid, smaller trees, and the repellent trees in the future. And this technology, combined with another set of technologies that we are developing. We have, for instance, a bait tree plant that we want to turn this plant as a natural insecticide for the psyllid to combine with the repellent and attractant plant. So we will have a combined action in field conditions. And at the end, using citrus trees that were found in the Oceanian several years ago that are from the same family, they can be cross-reacted with citrus, we are finding resistant genes against the bacterium. So the ultimate goal of this big biotech project is to have plants that are resistant for the bacterium that cause greening. Okay? So we would leave that for our discussion session. I thank all from the Citrus team, and I will be available for questions at the end.